I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them. So we have a new episode coming out about antibodies, and we're asking people, what is an antibody?
So it's a molecule that is produced by our immune cells to fight off pathogens.
I will say that it's something that really helps your body create the right defense system against any disease, anything that is attacking your body.
Yes, there's a monoclonal antibody and then there's bispecific, but I think in essence an antibody is something that's a part of the immune system that elicits a response. Yeah.
We know, but we are not sure about the absolute correct answer.
I can tell you how to get that antibody to the patient, but...
An antibody is a protein that is made by B cells that serves all kinds of functions in the immune system but can be used in drug development as a tool to treat diseases.
And if you could design your ideal antibody, what would it do?
I mean, a dream antibody would cure a disease, or nearly cure a disease, right?
I would like it to cure any type of cancer.
I guess to target the disease cells or the disease without harming the individual you're treating. Yeah.
I think an antibody that is customized to the individual or even can adapt to the individual's needs and the environment, I mean, maybe that's where we're heading in the future.
I'm delighted to be here today with Paul Carter and Yan Wu to talk about antibodies and antibody engineering. So Paul, what exactly is antibody engineering?
Apart from having as much fun as possible, it means that we try to redesign antibodies to do things, maybe the things which they do already, and we try to improve upon them. We try to get them to do things which they've never actually done before in nature.
So how did we decide and figure out how to use antibodies as medicines?
Well, this is something which has been a long time in the coming, really going back to the mid-1970s with just the pioneer work of Kohler and Milstein inventing the hybridoma technology. And so it was thought at that time that it would be a fairly simple kind of leap to go from being able to make antibodies to actually develop them as therapeutics. In reality, it took us as a field more than 20 years, and it was a bumpy road.
A bumpy road indeed. But before we keep going down the road, I'd like to get a quick definition. Remind me, a hybridoma is when you were able to take the cell from the mouse or the human that makes the antibodies, the B cell, and fuse it with another cell so that it could make them in a dish. Am I correct?
That's right.
So there's a lot of antibody discovery technologies. So originally the major one for many years are hybridoma. So the hybridoma technology is really the foundation to where we are using therapeutic antibodies. So hybridoma, usually you take the B cells from the mouse, beginning from the mouse spleens or lymph nodes, and fuse with the immortal myeloid cells, and basically form a hybridoma and continually secrete antibodies. And these antibodies are monoclonal antibodies, so the one single specificities.
Right, because an organism will make multiple antibodies. If you just get antibodies from the plasma of a mouse even if it's against one protein, they'll recognize multiple different, what you'd call epitopes, right, on the protein. But the hybridoma meant you got one single clone of an antibody that you knew exactly what it bound to. And drugs are monoclonal antibodies.
Yes.
Right? What was the first one that was ever used?
The first one was a molecule approved in 1986.
Yeah.
It wasn't very successful, and there was a period of about more than 10 years where lots of different groups would take mouse antibodies into clinical trials, particularly in cancer. Our result was really pretty dismal, that these were recognized as foreign proteins by people. People mounted an immune response which neutralized them, and they basically just didn't do anything. So that was a kind of a low point in the field.
I'm remembering some of my own antibody background now, because what you expect your antibody to do is target the immune system to kill the thing it attaches to, right? So that's why you can probably explain this better, that in cancer, an antibody against a cancer cell was supposed to bring, was it called ADCC?
Yeah, that's one of the
yes.
mechanisms.
Okay.
So one strategy with antibodies for cancer is exactly that, to try to recruit immune cells to kill the tumor cells. And that doesn't work very well with mouse antibodies and human cells.
Basically, you can say there is about variety, but the simplify is, let's say, block, flag, and deliver.
Okay.
So block, or use the specificity of antibody to recognize a unique antigens in the cell surface or secreted protein that block ligand receptor bindings.
Yep.
Right. So there's another way is flag it to see this is the highly expressed in the cancer cell. So you bring the antibody there, and you basically eradicate the cancer cells. And deliver is really using the antibody to deliver your payload to the site of action.
So when we think about an antibody in simple terms, we always draw it as looking like a Y, don't we? And the two tops of the Y, those are the parts of the antibody that recognize the antigen. And a typical native antibody, those two tops of the Y both recognize the same thing that we call an epitope. But I know that we've been developing, and there are drugs now that are called bispecific antibodies, which are really, really cool. So you make an antibody that would never exist in nature, that's got one arm of the Y recognizes one epitope, and another arm of the Y recognizes another epitope. So you can bring two things in the body in proximity to each other, which wouldn't normally be in proximity to each other with antibodies. Is that right?
No, that's right. So you can, like, say, binding a tumor cell and an immune cell, like a T cell.
Yeah. And then the other thing that I think is really cool is the ADCs, the antibody drug conjugates, where I think you stick something onto an antibody and it takes it to a cell, right?
It's a seductively simple idea to take an antibody, arm it with something which is, exceedingly potent and you couldn't use on its own. But the devil's in the details of the so-called payload, the so-called linker, how you attach it.
Yes.
How many of these you stick on your antibody. Yeah. And as a field, it kind of collectively took us many kind of iterations to get that to the point where it actually works.
Yeah. ADC has been worked very well, but it's still a challenging field.
What are the challenges?
The challenge is really the TI therapeutic index.
Okay.
Because you think you put antibody, have a magic payload, and can cure cancer, but there's a toxicity.
I think ADC is sort of an example of a technology which not only took a very long time to develop, but sort of went through cycles where there was no interest in the field, or very limited interest, to maybe just pendulums maybe swung too much in the other direction, to kind of irrational exuberance about they were going to solve everything. And then, so you go through these... sort
of pit of despair, and then you come back out the other side.
These cycles, so it's...
In science, in drug discovery, you have to start with the biology to really truly understand the pathway, the mechanism of action, the disease, right? And also you have to think about the clinical plan, see whether the molecule can be developed, but also combine with the technology innovations. So for us, we basically, in the past, we tried to make a lot of successful antibodies. So still, most of the drugs are still monoclonal antibodies. But right now, the field mostly is focused on make the undruggable targets druggable, or make the previous druggable targets with a totally new way.
Hey, Maria.
Hi.
Hi, Maria.
Hello.
So I heard ADC in there and I heard ADCC. What's the difference? What's going on?
Yes, I was confused by these terms many years ago when I first started to work in antibody drug development too. ADCC is a natural property of an antibody. It's called antibody-dependent cellular cytotoxicity. And so when your body makes antibodies against, an infected cell, that antibody binds to that cell, sends other immune cells to come kill that cell. So that's one of the things antibodies do. And when we use antibodies traditional antibodies to kill cancer cells, that's usually how they're working. It's like, the antibody's recognizing something on a cancer cell, and we're using the natural property of an antibody to its ADCC properties to drug that target, to kill that cell. You can engineer out the ADCC properties if you just want an antibody to go and bind to something and block it and not have this immune system activating effect. So depending on what you want your antibody to do, you would engineer it to have ADCC or not have ADCC. ADC stands for antibody drug conjugate, and that's when you tie a small molecule, usually a toxin, onto an antibody to deliver it to a cell. So it's a completely different thing that sounds quite similar.
So Maria, Jan was talking about undruggable targets. Is the word undruggable accurate? Is a target really not druggable?
Well, that's such a great question. I think it depends what you're trying to drug it with. I mean, for an antibody, the target does need to be extracellular, really. There's some places antibodies just don't go. But in the broader question of drug discovery, I think there are a lot of people now who are hopeful that there will be a modality that can tackle almost any drug target, and that we shouldn't be thinking about things as being undruggable. Some things are going to be harder than others. So Wellington wanted me to ask this question, which is, so we talked about bispecific antibodies, and he was wondering, is this like, disposable razors? We have 2 blades. Can we get 3 blades? Can we get to 7 blades? Is there like an extra strip on there? What else can we do with antibodies?
I think we can make more. So monospecific is a basic. Yeah. Now we're routinely making bispecific. So we're going to add in more modalities to it. So we're working on trispecific. I won't think we're going to do 7 specific, right? But we do have a variety of modalities. Doing fusion proteins, we can bispecific. Then there's fusing another antibody, a protein like cytokine fused to the C terminal of the antibody. Or we can link the modalities of antibody in we call it bead in a string.
Okay.
You can use your imagination. Of course, we have to have a follow-up with the technology that we can produce those complex molecules in a single cell that become the drug in a vial.
For sure you can make bispecifics, trispecifics. You can certainly add additional specificities. I think I don't know what the record is. I think it's at least 5. But the question you have to ask yourself is why. Every additional kind of binding site you add, whether it's binding the same molecule or a different one, adds complexity, because you can now tune each one. You can make it a higher affinity or low affinity or anything in between. And so there can be very good reasons for adding extra binding sites to being able to, say, enhance their selective binding to, say, a tumor cell over a normal cell. But it can certainly be overdone. You can make something which is extremely complex to make. And I can certainly, from personal experience, I've sort of worked on things in my, going back sort of decades where they were so complex, so-called immunoliposomes. And we got some very nice papers which are very widely cited, but they were kind of essentially useless in terms of benefiting people. They're just so complex, so difficult to make. And so it's shaped my thinking that you, in terms of how you develop drugs, that you have this kind of toolbox of technologies, and it's great to be pushing the envelope as to what's possible. But it also, I think it's kind of wise to be kind of grounded in pragmatism to make your drugs only as complex as they need to be to get the job done. And if you can do something with something simpler, other things being equal, that's probably going to be a preferred way to go.
Yes. In our experience, on the paper, you can design any molecule you want. Yeah. But the most successful drugs are usually the simple ones.
Paul, shall we go back to that bumpy road that you mentioned at the beginning?
So let's talk about the bumpy road. So we started working in antibodies in 1990, and I think their prevailing wisdom in many places, actually arguably most places, is that given this history of really no success with mouse antibodies, that people have kind of written off antibodies as ever being sort of useful. And so it was definitely an exercise in sledding uphill in terms of trying to kind of overcome the mindset that this would not work. And of course, we had no idea whether it would work either, but, we were just sort of young enough and pig-headed enough to pursue it anyway.
And philosophically, it should work, right? I think you must have had that drive.
Or just feeling that, with our advent of humanization, here's a technology that has the potential to address the known limitations of mouse antibodies. Of course, you don't know for sure until you get into the clinic whether it actually will. And even if it does, there may be some other obstacles that you don't even know about yet.
It must have been amazing when you first saw, was it human PK data or something that told you that the antibodies were okay?
I would say the first moment was when we were humanizing our first antibody. And within a few months, we had made a humanized version that worked almost as well as the mouse antibody in the test tube. And then a few months later, actually had a version that we felt was, based on our knowledge at the time, was as good as we could make it and was kind of worth our exploration in the clinic. That was kind of a very, very exciting few years.
Amazing.
Yeah, truly, antibody has been described as a magic bullet a very long time ago, until the technology innovation really make it possible to be a real therapeutics. You think about from hybridoma, mouse antibody, to chimera, to humanized antibody. Now we have actually ways to making fully human antibodies.
When we talk about design of molecules, I wanted to ask you a question that we're asking everybody this season on the podcast, which is about how AI and machine learning are applied to the work you do. And this feels like a perfect place to talk a little bit about how that's applied to antibody and protein drug design.
I think that's a great question. This is almost kind of feels like a gold rush era in terms of applying AI to redesigning proteins. And for sure, it will have an impact. It's starting to have an impact. But exactly where it's going to be most useful, I think it remains to be seen. I think the sort of the holy grail is to be able to design our antibodies and proteins from scratch that bind at specific sites, specific places, and other proteins. That's a very difficult thing to do. I think there are perhaps simpler problems which are more tractable. And one of the things that we do in engineering antibodies is, to use a bit of jargon, would be sort of multi-parameter optimization, which is a fancy way of saying you want to maybe engineer an antibody to do several things at the same time. So maybe have high affinity, have good biological activity, have other drug-like attributes. And so I think their AI has at least the potential to be able to simultaneously solve to satisfy all of those things. Whereas, historically in the antibody field, we've tended to do one thing. So we've tried to improve the affinity. And then sometimes we find out in improving the affinity, we've mucked something else up, and then we have to go back and fix it. And then we've mucked something else up, and we have to go back and fix that. And so rather than do things serially like that, if you have enough capability in AI in terms of, sort of models, in principle and probably in practice over time, you'll be able to improve one property and maybe improve other properties at the same time, or at least not mess them up.
I think with the advance of computation, machine learning, and artificial intelligence, may be a game changer for antibody discovery and engineering, but it will be gradual work.
Yeah.
Because the machine learning AI needs a lot of data.
I see.
Although we have over 100 approved drugs, antibodies on the market, but there are not that many.
Yeah.
So we actually need working closely, side by side, with machine learning AI scientists. A lot of industry too, including us, is using a model called lab in the loop. So basically, we're closely working with the machine learning scientists, with the bench scientists. Bench scientists provide as much data as possible and help the machine learning scientists train the model. And when we also need a test in the wet lab for the design the molecule to see whether it achieves the goal. But I think in the future, it's looking very promising, So we need to continue to work on now. Everything takes time for the technology development.
I think it's certainly an exciting time in the field, and they're very kind of different disciplines. It's like kind of learning a foreign language if you're not an AI person, trying to understand the rudiments of that. And it's also for the AI folk, learning some of their, acronyms and their ways of the wet lab world. It's kind of like you're kind of trying to get people or train people so that they're bilingual. And so maybe experts in one of those disciplines, but have enough understanding of the other discipline can ask good questions. And I completely agree with Jan. The need to do this, iteratively, and to do that so-called lab in the loop as fast as possible, because that's how we're going to make progress. AI is definitely an endeavor where, other things being equal, the larger the data sets that you have, the more that is possible. So I think it feels like this early period is trying to learn the language of the other discipline, and together figuring out what kinds of problems are most tractable. Because you're just kind of only limited by your imagination in terms of things that you can do. But there are some things which aren't really feasible at the moment because it just takes us too much time and effort. We just don't have enough data.
Yeah, it's exciting. I think the only way to be very successful is working very closely between the antibody engineers and the machine learning scientists, working as one team.
Yeah.
Hey, Maria, they were talking about lab in the loop. What do you know about that?
So the way I understand it, it's the idea that human physiology is incredibly complicated. The inner workings of the cells are incredibly complicated. And what we typically have done is kind of approached it piecemeal, like here's this kinase cascade, or here's this receptor signaling pathway, let's drug it, let's look at it. And the idea of lab in a loop is that you generate an enormous amount of data, whether that's from cells or from treating animals or even from patients, and you crunch that using AI and machine learning to see how the drug that you made manipulated the whole system, like in a way that you wouldn't be able to understand just with your just by looking at things in isolation. And then you use that information to then help you feed back and design better molecules or choose better targets for drug discovery.
Maria, when Paul is talking about the humanization of antibodies, what is he talking about?
Oh, so the first antibodies that were made to use as therapies were made from mouse B cells. So they had a mouse sequence. So when you would put them into a human being to use as a drug, the human would have an immune response against the mouse protein. So they had a lot of side effects that they didn't work very well. So because there was at the time no ability to make human antibodies from human B cells, you had to take this mouse protein and make it look so much like a human protein that the immune system of the patient wouldn't react to it, essentially. So it was a way of replacing sequences in the antibody, so you don't lose any of its functionality, but it looks human enough that it is safe and well tolerated. It's interesting that there are only a hundred or so approved drugs, which I hadn't thought about it that way. That does limit the data set of what a successful antibody drug is. So what other limitations are there around the data availability for the work that we're doing with AI and machine learning?
I think the most challenge is actually good data. You really need a good data to train the model. It's called garbage in, garbage out. You have a lot of data sequences, but you really need to say the antibody with the right sequence, with the right affinity, with the right binding site. So all these data, binding, pharmacokinetics, and its developability. So these are all really need a real data, a high quality data to train the model. So we actually work together to see, we already accumulated a lot of data over the years, but I think we actually need to generate more data, high quality data. But also we need, as Paul said, we need to ask the right question. So basically, after we get to know each other, we really ask what's the most fundamental question in antibody engineering that we think machine learning AI can contribute. In the short term, antibody optimization. But in the long term, it's more like de novo design.
Yeah.
Right now, it's really need to work still in the model as a lab in a loop with a computer design and wet lab.
I think something else that's important to really be rigorous in this area is when you're looking at AI designs, compare them with human experts. And obviously, depending on the particular experiment, maybe you just want to, randomly select, related sequences. And that will kind of keep us honest as to whether we're being successful or not. And so that I think is kind of an important paradigm in exploring to see what AI actually adds.
So being kind of rigorous about it, to say, well, I'm not going to assume that this is going to give me something
No,
better.
because otherwise if it works or doesn't work, maybe that just happened by dumb luck.
Yeah, right now it's basically the data generation, training the model, and validation.
Yeah.
The goal is eventually to improve the process and speed of discovery and engineering.
Yeah. So before I let you guys go, I wanted to ask you, what are you excited about right now in the field of antibody drugs?
I can go first. We kind of talked about the low-hanging fruit analogy, and you think of, okay, what are the challenges now that we really don't know how to do or can't do very well? And I would certainly include things like, getting antibodies into the brain in useful amounts. We talked about sort of gut delivery. Get excited about targeted delivery, so trying to get antibodies to be functional where you want them. So active for binding or maybe other activities in a tumor, but not at other places. And so I think there are lots of challenges there for their antibody engineers to continue to work on for many years to come.
I agree with Paul. Basically, we try to continue solve the challenging disease problems. So we need to continue to do our technology development with the new modalities, new ways of delivery. So for example, we really try to work on a new format, modality of antibodies, probably combine the antibody large molecule with the small molecules, with antibody, with the peptide, right? To see how do we enhance the new mode of acting. It's not just what one antibody can do, it's go beyond one antibody can do. So I think there's a lot more we can do.
It's a pretty exciting time.
With the continued technology development and the new modalities.
Fantastic. Thank you so much for sharing your enthusiasm for this really exciting field.
Thank you so much for having us.
Yeah, thank you.
Maria, that was a great episode.
Thank you.
What is your relationship to antibodies, antibody engineers in your career?
So early on in my career, I led a group that was looking for novel drug targets for antibodies for metabolic diseases. So I used to work very closely with antibody engineering scientists. Like my job was to say, hey, why don't we design an antibody against this, cell surface receptor, because maybe it will help improve fatty liver disease, for example. And then I would get to work with the scientists on designing the antibodies, testing them, that type of thing. So I've part of my career, a good 3 or 4 years, was spent doing leading teams doing antibody drug discovery programs.
So Maria, you were asking this at the beginning of the episode, but I get to ask it to you now. What is your dream antibody?
Oh, that's such a great question. So I would love to see, I think going back to our episodes we've done on Alzheimer's disease, I think the idea that you could get an antibody that can get into the brain, that we can deliver in a nice, easy manner to a patient, that can really stop the progression of that disease by clearing some of the proteins and things that we know are involved in Alzheimer's disease. So I see a lot of promise for antibodies in the CNS disease area space. But there are so many exciting applications, I think, for antibodies going forward. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It will help new people to find us. And be sure to subscribe. If you have a question about the show, you can contact us at podcast@gene.com. That's GENE.com. And now for me, it's back to wrestling with data.